The glycolysis pathway, the metabolic process that breaks down glucose to produce energy, requires the conversion of fructose 6-phosphate, F6P, into fructose 1,6-bisphosphate, F16BP. An enzyme known as phosphofructokinase 1, PFK1, is responsible for catalysing this reaction. Phosphofructokinase 1. PFK1 must be triggered in order for the conversion to take place. The levels of ADP, adenosine triphosphate, ADP, adenosine diphosphate, and AMP, adenosine monophosphate, as well as citrate, an intermediary in the citric acid cycle, all play a role in the regulation of this enzyme. Citrate and high ADP concentrations restrict PFK1 function, whereas AMP and low ADP concentrations promote it. The amount of ADP in the cell affects the activation of PFK1. High concentrations of ADP limit the activity of PFK1 because they function as a negative allosteric regulator of the enzyme. Binding to allosteric site. The enzyme PFK1 possesses an allosteric site, which is a specific spot where it can attach to control molecules and modify its activity. ADP molecules bind to the allosteric region of PFK1 when the concentration of ADP is high. Inhibition of PFK1 activity. A conformational shift in PFK1 brought on by the binding of ADP to the allosteric site results in its suppression. As a result, fructose 6-phosphate is converted to fructose 1,6-bisphosphate at a slower rate and the enzyme is less active. Regulation of glycolysis. A regulatory mechanism to manage the pace of glycolysis is PFK1 suppression by ADP. High ADP levels are a sign that the cell has enough energy reserves. Since more glycolysis of glucose is not required in this situation, PFK1 is blocked to slow the process and save energy. AMP and ADP effects. Conversely, PFK1 is negatively impacted by high quantities of AMP-ADP, and low levels of ADP. A different allosteric site on PFK1 is occupied by AMP and ADP, which promotes the enzyme's activity. This guarantees that glycolysis is increased to produce more ADP when cellular energy levels are low. Next is binding of fructose 6-phosphate. After becoming activated, fructose 6-phosphate, the enzyme substrate, attaches to PFK1. The first stages of glycolysis produce the sugar molecule F6P from glucose. ADP utilization. The binding of F6P to PFK1 permits ADP to bind to a specific location on the enzyme. Then, ADP undergoes hydrolysis, losing one of its phosphate groups and changing into ADP. This ADP hydrolysis reaction supplies the energy required for the working steps of the cycle. Formation of fructose 1,6-bisphosphate. A phosphate group from ADP is transferred to F6P as a result of ADP hydrolysis, creating fructose 1,6-bisphosphate. The original phosphate group from F6P is still linked to carbon number 6, while the phosphate group is attached to carbon number 1 of the fructose molecule. As a result, the substance is known as fructose 1,6-bisphosphate. PFK1 inhibition. The production of fructose 1,6-bisphosphate acts as a feedback inhibitor for PFK1. When energy needs are met, PFK1 activity is inhibited at high concentrations, preventing an excessive buildup of downstream glycolytic intermediates. In conclusion, PFK1 activity is stimulated by high levels of AMP and ADP and inhibited by high ADP levels. By controlling the rate of glycolysis in response to energy demands, this process aids in the maintenance of energy homeostasis in the cell.